Largely, we use biomarkers, I'm in oncology, as a method of selecting patients, the right patients for clinical trials. Since drugs tend to work well in some patients but not all, we need to know how to direct the drug to the right patient to get the desired outcome for therapy. So a large part of my career has been designing biomarker assays to tell us whether or not patients are sick, how sick they are, and whether or not they will be helped by a particular form of therapy. Technology plays an enormous role. It has changed so much over the past couple of decades, from things that are fairly easy and old-fashioned and won Nobel Prizes 50 years ago to now we have the ability to interrogate the genome at a scale that people imagined 20 years ago, but we're doing it on a daily basis. We have more information, literally, than we know what to do with. We can't make sense of it all. The technology has allowed us to probe deeper, further, faster, and into things that we were not able to visualize before. The big hope from all that now is, with that technology improving by leaps and bounds, can we take that and turn that into a paradigm where we can actually better help the patients? Because this is what it's all about. We need to understand the biology behind the disease. We need to understand what applies to that particular patient, and then whether or not the drug of choice will actually benefit that patient. Reactive medicine is really, we've seen something, and so we know it's important. And proactive medicine is where we are in the biomarker field now. We know there have to be important things because we know we don't understand everything about the disease, and we have to go find out what are the important parts of that. So that also goes back to playing in the technology arena, because we now can examine more things for more information to let us know exactly what might be important. So we look at all different kinds of biomarkers. A biomarker can basically be anything. It simply is something that tells you something about something. And if it tells you something useful, you can put it into a clinical trial and benefit patients. So this is, instead of a one-size-fits-all approach, as the saying goes, it's individualized therapy. And that's important because there are very few places where one size does fit all. It does happen certainly in a number of different fields, but not everywhere, and not completely within each and every given field. And in cancer, we have certain disease states that we have very effective drugs that have 95, 98% cure rate, which is wonderful. Our current flagships in immuno-oncology are about 25% effective. And we know we're doing great things for those patients who are helped. But that still leaves us with 3/4 of the patient population that we can't do anything for. So how do we deconvolute the disease down to the individual patient level? We know everything about the population. We know how many people will respond, how many won't. But we don't know which ones. And that's where it matters, because we're not treating a population, we're treating an individual patient. And the biomarkers are the thing that can tell us which patient we should treat and we should not treat. And now we're trying to determine what are all the niceties that Mother Nature has put into cell growth that over the past million years or so that we don't quite understand, because when we treat people and we go with the biomarkers that we have at hand, we don't get perfect results, and we want to do better. We need to do better. There's an enormous amount of effort, time, money that's put into this, and we know we're not curing every patient that comes into the hospital for treatment. And I think that's the end goal of the game. We are finally at the stage where we can take a look at almost everything that goes on in a cell or a tumor. And it's really a matter now of learning how that impacts. We had the promise of the Human Genome Initiative that would solve all our medical problems 20 years ago when it was done. It hasn't happened yet because it's a lot more complex than that. But we are learning now very rapidly how to tease apart all of the different systems in a cell that interact with each other to produce this. And it's that kind of thing that is really the cornerstone to this. Biomarkers tend to be physical components, a protein, a molecule that we measure, and that's very easy. But that's not what the final clinical outcome is. It's an amalgam of all the different biomarkers, the DNAs, the RNAs, the proteins, the lipids, everything else that comes together to produce a clinical effect. And it's that clinical effect that we have to understand. And we are much closer to being able to do that than we have been ever before.